Hello. How are we all doing? I've already lost my YouTube feed. Okay, so Tuesdays, 5 o'clock, I'm going to be here with you doing your A-level biology. Basically, any questions you've got, you can bring them to me and I will answer them. We're going to go maybe through some past paper questions. I'm going to show you some exam technique. If you've got any issues with what's going on with your world of A-level biology right now, let me know in the chat. Basically, we can I want to be as useful to you guys as I possibly can. So let's just find this over here. Okay, wonderful. So yeah, let me know what's real for you, basically, in the world of A-level biology right now. There's a question that I've got lined up that maybe we can go through, but most of all, I just want to be helpful for you guys. So those of you who are my regular subscribers, I'm doing my full web class starting at 6 o'clock. We're actually doing a slightly different format for those of you who are part of the Ultimate Guide. I'm doing AQA questions, so like AQA specific questions from 6 till 6:45, and then OCR specific questions from 6:45 till 7:30, is it? Yeah, 45 minutes on each roughly, but there'll be some mix over. I will be answering questions specific to each exam board during each of those sessions. You're welcome to attend the other sessions, but yeah, I will only answer in the context of the exam board during that 45-minute window. Hey everyone, hey Nat, hi Pierre, hey Kieran. So, Kieran, these YouTube lives are all on the Taylor Tutors YouTube channel, so you can effectively join us. You can watch the recording back, you'll be able to see all the recordings and the history, so just find them on the Taylor Tutors YouTube channel, basically. Nat, I'm going to be doing basically most of A-level biology is pretty simple. So I don't know the Cambridge exam board. I wrote my courses for AQA, OCR, Excel. I know the WJ. I have taught Cambridge, but that was the old spec some time ago. So Cambridge is most similar to the OCR specification of the big ones. I mean, so few students do that. It's not very well sat in terms of numbers. Is this tomorrow? What is what tomorrow? So it's Tuesday. It's Tuesdays 5 until 5:30. I'm just going to be doing some drop-in biology question help. So let me just pull my chat a bit more underneath, so I'm looking vaguely at the camera. Oh, no, jumped onto my other screen. I can cover a bit of both, Hillary. Yeah, I'm going to be doing. Any questions that you have. What are your biggest problems? Like, how's revision going? Are you still on Easter? Have you finished Easter? How do you feel in terms of your motivation right now? Are you super amped? Are you like stressed out and you're just like working all the time in your Easter holidays? Or did you feel like Easter was your last chance to relax a little bit and so you maybe didn't do as much work as you might have planned to do? Okay, gains. So design and experiment questions. Do you do Edexcel? Mostly they are the Edexcel questions, but they do come up under all the examples. Okay, so let's just tackle that for a second. If you want to design an investigation into, I'm not even going to pick a topic. What are the things that you need to know or you need to include in any investigation? Now, let's say it's a 6-mark question. 3 of the 6 or more are going to be generic to all practicals. So what could they be? Let's brainstorm some of them. And wherever those, I want you guys to be generating the content. I want you to be thinking about it. Just passively listening to me and not taking notes is not really going to do any like actual benefit. Okay, so AQA is quite rare to find out questions on the nephron can't be found in new specs. So yeah, there's loads of kidney and nephron questions. So don't worry, there's a lack of questions. WJ, OCR. Edexcel don't cover the nephron, so don't look in any old spec or new spec Edexcel papers for that, but you will find that. The loop of Henle, I can maybe find a question on that if you'd like to go through a loop of Henle question. I got asked lots of great questions on my course this week about the loop of Henle. I've spent quite a lot of time answering those questions. Courtney's not achieved anything over Easter. When do you think the stress is going to kick in and like actually get you motivated to do some work? Yeah, all the 2018 papers will be released on the 4th of May. So yeah, the revision kit is going to include all of that, but we can't, we've been asked by schools not to publish them until then, and the exam boards don't publish them. Well, Edexcel have published theirs, but we've been asked by schools to hold them back so they can use them for mocks. Okay, so Gainesville Guides. what do you want to keep the same? If it says to design an investigation to test, but it doesn't matter what the thing is. Things that we can talk about. Pierre, control variables. Obviously, which variables you want to control so that it's a valid test of what you're testing. So you temperature, and also you often have to include a method as to how. You can't just say control temperature. You might say keep the test tubes in water baths at temperatures. You want to be using a range of at least 5, depending on what you're investigating. Let's say at least 5 different temperatures, and then give a suitable, suggestible range. So that might be, if you're looking at enzymes, obviously you're not going to do from 1 million degrees to 2 million degrees. Just simply no. But it's looking, if it's asking you to describe how you do the method, then you need to say use a range of at least 5 different temperatures, e.g. 10-degree intervals between 5 degrees and 70 degrees. Something like that. Okay, what else might you want to say? You want to record, you want to do at least 3 repeats and take mean values and perform a statistical test. Obviously, the statistical test will depend on the type of data that you are collecting. But again, if you know which type of test, stats test you should use for each one, then that's a pretty easy, low gain, easy gains for those marks. Right, what else would I say in terms of those? You want to, take at least 3 values, minimum 3 values. You want to say a bit more. Take a mean so you can identify anomalous results, perform a stats test, control your variables and tell them how you're going to control your variables. So that alone is probably going to pick you up 3 or 4 marks on any of those describe a practical questions. Okay, so let's have a look at some questions here. We've got another 23 minutes. I've got to prepare for my ultimate guide students who I'm seeing at 6 o'clock. Biggest problems are applying knowledge to questions at AO3 part. So Navsin, what have you been doing to practice your AO3 questions? What have been your techniques that you've been using to improve your AO3 parts of the spec? Courtney saying motivation has kicked in. Oh, it kicked in and then you lost it. Okay. You're done with 2 or 3 past papers and for AQA and couldn't find any more, so you started other topics on these papers. It depends what you mean. There's obviously paper 1, paper 2, and paper 3. There's the specimen 2015. There's actually a couple of other set papers for those, but again, they're not. We've been asked not to release those. But there's the 2017 and the 2018 papers. There's 3 papers, 3 years. And so there's 9 past papers for AQA that are available to you to revise from. So that's 9, and that's like, if you want it, and you should be doing each past paper a minimum of twice, but ideally 3 times, maybe even more. So that's, if you're going to do 9 papers 3 times, that's 27 time conditioned closed book exams. That's a lot, and they're 2 hours each. So that's like 50 hours. Plus, then when you find your holes in your knowledge, you have to go away and go and improve those bits and pieces of the questions you didn't do very well on. That's a huge amount, and that's enough past papers. Trust me, that's enough. That's definitely, I'd say, enough. In photosynthesis, why can't NADPH be produced in cyclic photophosphorylation? I could draw that for you. That's probably going to be the easiest thing to do. How am I actually going to share my screen? I'm going to have to use quick time on that. So let's pull up and I'll do a little demo of that for you. Who asked me that question? Let's import. In fact, let's just go to create a new page. My screen brightness is definitely not bright enough here. There. Okay, so let's do a little bit of cyclic analysis. So this is year 2 photosynthesis. Very quick explanation as to why cyclic can't produce NAD, reduced NADP. Oh, spinning wheel of death, never a good sign. Okay, sweet. So I think it's going to be in here. I'll just share my iPad screen with you. Okay, so in photosynthesis, we have our thylakoid membrane. Well, that pen, I was not using this last. Okay, we've got our thylakoid membrane. Ordinarily, you're going to have photosystem 2, photosystem 1, and then we're going to have our ATP synthase. Oh, which way around does our head go? I think our head goes on the inside, but you're never going to get marked down for that. Okay, so that's our ATP synthase. We're going to forget photosystem 2 for now, because cyclic photophosphorylation doesn't involve photosystem 2. So photosystem 1, we've got our chlorophyll, which I may as well draw in green. Light energy is going to strike chlorophyll, and it's going to excite an electron and raise it up to another energy level. But this normally, if this was, in fact, I'm just going to do cyclic first. This electron is then basically going to flow down through the membrane through decreasing energy levels through these various different proteins. These are electron carrier proteins in the thylakoid membrane, and this is an electron transport chain. But the electron, if we look, the electron just returns to where it came from, just does this loop. And the electrons flowing through the electron transport chain provide energy to pump protons from the stroma into the thylakoid. But that's all it does. So we build up this nice proton gradient. I should probably say where we are. This is inside the thylakoid. And this is in the stroma. And obviously it's photosynthesis takes place. This is the thylakoid membrane here. So now we've got our proton gradient, then our protons can diffuse through ATP synthase, down their concentration gradient, and that energy is used to join ADP and inorganic phosphate to make ATP. So I can't see the chat at the moment. Let me just go back to the screen where I can see the chat. No, window is not the one that I want. Okay, so why can't we make reduced NADP is my question to you. This is cyclic because this is going in a cycle. The electron is just going round, round, round in circles. But that's exactly the reason why I can't make NADP. Because in order to make NADP, if I do non-cyclic photophosphorylation, then light energy is going to strike this. It's going to raise the electron to another energy level. It's going to flow through the various different proteins in the thylakoid membrane. It's then going to come to this, and then it's going to absorb more light energy in photosystem 1. But in non-cyclic, this electron is going to leave the photosystem. So it's basically because we don't have this electron here. So this in non-cyclic, you get this electron and this proton, and we get NADP plus an electron plus a proton, and that's going to give us our NADPH. Yeah, but we must have this electron here. But in cyclic, this electron stays here. So without this electron being outside of the photosystem, it's not possible to make reduced NADP. It's missing one of its ingredients. So that's why non-cyclic A. So the only product of non-cyclic photophosphorylation, of cyclic photophosphorylation is ATP. Okay, so let's stop my screen sharing. I'll come back to you over here. Okay, let's see if I can just catch up on some questions on my own. Pierre, I know you, so yeah, if you register for the web class now, I will accept you just before the web class, and I finish this at 5:30. Rebecca, the web class is our Ultimate Guide students, so if you want to do those, you need an Ultimate Guide subscription. No worries if you can't be web class Courtney, that's no worries at all. Your teacher hasn't probably taught you how to answer AO3 questions. You don't know where to write. It sounds like you need a revision is what you need. That's exactly why I wrote the course, because students and teachers are often not covering that exam technique very well. The difference between describe and explain. So describe is a description of, do not give reasons. The graph goes up, the graph goes down. You do not have to say why. Describe the process of, this happens. Explain means give a reason for. So I just remove the word explain from my head and I go with give reasons for. And that makes it way easier to understand what the question is. So why does the graph go up? The graph goes up because there's more kinetic energy, there's more enzyme substrate complexes being made, whatever that graph might be doing. 25 markers, find past answers. Yeah, there's basically all the past essay titles are in the revision kit if you want to have a look at them. and attempt to answer them. Yeah, so like a good detailed plan. Yeah, that's good advice, Courtney. I'd say definitely so. Can upload a video going through a year paper? I don't quite understand. Can you put that as a question, and I will answer it as a question for sure. I don't know, is it ICRA? ICRA, I have recorded myself answering all 27 of the new spec papers, AQA, OCI, Excel, 2015, 2017, 2018, for paper 1, paper 2, and paper 3. That's all 27 papers that I have done. That's what the revision kit is, part of the revision kit. What advice do you give to answer the maths questions? Okay, so Hiba, great question. The good news about the math skills questions is that you know 10% minimum of every exam that you do is going to be on the math skills, 10% minimum. So they, compared to the rest of the course, where you get, let's say, a 30% of everything else you learn comes up, a solid proportion of the things come up in the math skills. So and they're skills, they're not knowledge. So you can practice them and practice them and practice them. So find all the maths, new spec maths questions from AQA, OCI, and Excel. The maths contingent is the same for all the exam boards. Go and find all those questions and practice the hell out of them. You know once you've done 20 percentage change questions, you know how you calculate percentage change. Once you know how to calculate ratios, you can do like lots and lots of repetition and practice. So definitely yes. That's basically the best way of doing it, or find the questions you are consistently getting wrong, whether it be volume of a sphere, whether that's, you need to know things like surface area calculations. There's some really devilishly tricky ones out there. But again, if you are well drilled and you're well prepared on all those math skills questions, you can basically walk into the exam knowing you're going to get 10% because you've practiced them. Logarithms are annoying. Definitely, log E. I've seen one log E question pop up recently. They're nearly always log base 10. But you need to know how to use your calculator in order to use logs because it's in the specification. Don't answer the math questions. I wouldn't say don't answer them, Courtney, because you will lose 10% of the marks. Where can you find them? In the revision kit. ICRA is where, like, if you want to see me answer those questions, that's in the revision kit. tailoredtutors.co.uk, go check it out. There's loads of stuff in there. How much to shave my mustache off? No price. No, not happening. Is there any point doing the old spec papers? I would definitely do the new spec papers from the other exam boards before you do the old spec papers. If there's a weak topic that you know you need more questions on from your exam board, that's when you delve into the old spec papers. If you are weak on tissue fluid formation and return, then you just can't find enough questions, then go into the old spec and download every single one of the past papers and search through them and find those questions. It's very, very time consuming, but without using the course that I wrote, I don't know any other way you can do that. If you have a Mac, it'll be slightly quicker because you download the folder, so it's through that way. But yeah, basically, that's what it. Definitely do questions from the other specs, but only after you've done your exam boards, all your exam board questions. Obviously, go with them first. And you've got paper 1, paper 2, paper 3, 2015, 2017, 2018, and you should be doing them at least twice, if not 3 times. That's 27 past papers. Now, when you've done that, when you've done each of those papers at least 3 times, then you can come back to me and say, I need some more questions. Because, like, really, you shouldn't be thinking about anything else until you've absolutely nailed all 9 papers and you've done them at least 3 times, so 27. Logarithms are in every single specification, Anna. Yeah, all the maths is set by Ofquat. There's basically no difference. There's tiny differences in the stats, but the maths is all the same. If you want to ask more extended questions, what's the best way of doing it aside from comments? In my course, Naomi, aside from the comments in the course, bring them to the web class. How do you mean? What kind of questions? Is it like based on a past paper question and submit them to a web class and I'll break them down and go through them in a web class with you? If it's in comments on my course, obviously you can ask me questions on any one of those. Depends what the kind of question is. More extended questions. Give me some more context, Naomi, and I'll tell you. Best way to revise paper 3 essays for AQA, plan loads of them. write quite a few under time conditions, but it's more important. Planning them is a great revision strategy anyway. Closed book, just like give yourself a question and like put down your plan in detail so you know exactly what's going to be paragraph 1 and what's going to be the details in it, paragraph 2 and what's going to be the details in it. I'm not going to give you all my secrets because I've written a full course on how to plan the entire AQA essay. And so, yeah, if you want that, then it's inside the revision kit. Is there a story behind the mustache? No, it's mostly my ex-girlfriend. Like, I was traveling with her after we'd been long since separated, and she didn't like it, so I kept growing it, basically. Describe it, explain, suggest, and design investigation. Any others that come up? There's lots more command words than that. Yeah, heaps. A lot of the AO1 ones you don't need to go, like state, name, give, calculate's actually AO2, but those are quite obvious. You have evaluate, but you have to do different things. If it depends on your exam board, and my advice that I'd give you for different, depends on your, for evaluate questions. Sophie, serial dilution calculations. Okay, so the way that serial dilution calculations questions are going to work, where are we at? 10 minutes to go. Okay, let's just share my screen over here. Let's go a new page. So basically, they're going to say you've got, in fact, I'm not drawing it will be easier if I draw this as like some kind of beaker. Oh dear, I can't draw a straight line. Okay, so you've got a beaker. It's got loads and loads of living organisms in it, typically. And then you're going to take out a fixed volume, let's say, I'm just going to make this up, 2 centimeters cubed of the solution. We don't know how much in there, and we're going to put that, let's make it 1. It'll be way easier to teach you if I make it 1 centimeter cubed. And you add that to 9 centimeters cubed of water, and you mix it, and then again, we basically repeat that process. What's our dilution factor here? Can't see the chat, but I'm assuming you're all going to know. Okay, so, and then this one, we take out, what do we want to take out? Let's take out 5 centimeters cubed, and we put it on a plate, and we culture it, and afterwards we see this many colonies. How many have we got? We've got 1, 2, 3, 4, 5, 6, 7, 8 colonies. 1, 2, 3, 4, 5, 6, 7, 8. Okay, so each one of these colonies was a single cell that we took out from this here test tube. So there's 8 colonies. Is everyone with me so far? Let me go back to why are you not showing me YouTube in the chat? Okay, here. Is everyone with me? Naomi, I'll come back to your question in a second. Okay, so we've got 8 colonies, and we've taken, so now we're basically going to have to work our way backwards to say how many, and let's say there's 15 centimeters cubed of the original solution. How many bacteria are there in the 15 centimeters cubed is the question. That's basically what most of them are going to come up with serial dilution. Well, our dilution factor each time here is 10. This is diluted 10 times, this is diluted 10 times, this is diluted 10 times. This isn't diluted, but we had 5 centimeters cubed of it. So in 5 centimeters cubed, we had 8 colonies. So what we want to know is how many is there per centimeter cubed in this test tube, which would be 8 divided by 5, gives us how many, this is bacteria per centimeter cubed in this test tube. And then we need to times that number by 10 to go back to this test tube. And then we need to times this number by 10 to get to this test tube. And then we need to times this number by 10 to get the original concentration. And then that gives us, that was 1 centimeter cubed. So we then need to times it by 15 to work out how many bacteria were in total were in that solution. I've brought my calculator in, which I didn't think I was going to need, but good job I did. So 5 divided by 8 gives us 1.6. I'll put the numbers down so it'll probably help you follow it through. So 1.6, and we're going to times by 10, times by 10, times by 10 gives me 1, 2, 3, yeah, so times by 10 times by 10 times by 10 gives me 1,600. And now that was in 1 centimeter cubed, so I now need to times by 15. Oh, that's a 10, not a 15. And you get, and it usually say give you answer in standard form because I haven't done as many dilutions as they normally do. So this is 24,000 bacteria. So this would be 2.4 times 10 to the 1, 2, 3, 4. Boom. And there is our answer. Does that make sense? Hardy Weinberg, oh, we'll see how we've got 4 minutes. Let me answer just some of those questions. I know Naomi, who was asking us what was going on. Identification from gene technology, that's too big of a question to delve into right now. Last year's AQA paper 2, what was the advantage of iPS stem cells over gene therapy? Well, it depends on, well, I mean, they're generally going to be longer lasting. Anyway, let me answer some other questions first. I'll maybe pick a quick Hardy Weinberg. Is it mainly year 2 topics that sit in A2? It depends on which exam board you are. Okay, you're struggling with how to use a cheat sheet, for example, how to incorporate them into your revision. Like you've got a stack of notes, but how do I learn them all? They're probably pretty good for just testing. I would use them to test your AO1 recall. So you could like make, like, what's your topic? Let's say it's speciation. So like the 6 things you need to know about speciation. If it's allopatral speciation, so geographically isolated, there's no inbreeding between the populations. Variation exists due to mutations. Different environmental conditions cause differential reproductive success. So there's a change in area of frequency over many generations. Okay, it's maybe slightly more than 6, but they are the bullet points you need to know. If you get described allopatral speciation, they're the things you have to write down. It doesn't matter which exam board you are, they are the rules. So like you want to be prepared for them to be able to describe the process of succession, colonization by a pioneer species, changes in environmental conditions, e.g. water availability, soil depth, or increased humus in the soil. It's outcompeted by another species. This continues over many generations until you reach climax community. Like, so if you need to be able to basically bang out and describe the process of, because the AO1 content is going to be 30% of your exam. 70% of your exam is going to be the AO2, AO3 application stuff. So that's going to be your practical skills. That's going to be analyzing data. That's going to be calculating rates. It's going to be explaining why something is happening, not just describing what is happening. So you're struggling. So that's what I would suggest, Naomi, is that you just basically set yourself little factual recall tests so you can get your factual recall, your AO1 content knowledge like sharp. I mean, ideally, I would get my students to be the point where you can do all of that by now, and then you've got, ideally, that's sort of April territory, and then you've got all of May to be like, okay, past papers and fine-tuning exam technique and revising all those bits and pieces. You're welcome to the cheat sheets in the past paper library in my course. So you can always ask me questions like that in the past papers course, or wherever would be most relevant to that. So I'm happy for that to be done. Okay, so dilution factors times 10, you nailed it. I'll do a very quick Hardy Weinberg. Analyzing data. Yeah, practice. They are very similar. The context of every question is going to be very, very different. But practice, 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 because the skills that you will need is a skill. It's not knowledge at that point for those analyses. So do them, mark them, analyze where things went wrong, and then repeat the same question. Repeat the same question. That's why it's so important to repeat the same exam papers, because unless you're improving on the questions you've already seen before, when you get a new question, the context is different. You can't see the pattern. You can't see what you're not doing. That's why you have to do the same question that you got wrong yesterday again and get it wrong again until you don't get it wrong anymore. And then when you do the next ones, you can apply that to the new question. It's a slow improvement. That's why you can't leave, if you want to get maximal gains, you can't leave it super last minute, because those are skills, and it's like they're skills that you should have been practicing for 2 years, but most teachers don't do it that way. Okay, I'll do very quick Hardy Weinberg and then I'm going to go. Finn, if you haven't been taught anything properly, get the ultimate guide. It will teach you everything you need to know. Okay, let's just go one more over here, share my screen. I'm going to just make up a Hardy-Weinberg question because the two equations we've got is P plus Q equals 1, and we've got P squared plus 2PQ plus Q squared equals 1. So if I said it's always going to be a single gene with two alleles, you're never going to get anything other than that. So let's say you've got tall and you've got lower T is dwarf, and it's recessive. So if 26% are okay, if 26% are, oh no, this is going to take you too long, that's too complicated. I'll do a simple one. Are dwarf. How many are heterozygous? So heterozygous is 2PQ. What do we know? This is the first question you've got to ask yourself, is this a genotype that I have, or is this a phenotype that I have? So like, which one is it? Yeah, Sophie, Tuesdays, 5 till 5:30, this is where you're gonna find me. Follow the YouTube channel and just put it in your diary. Obviously, if you want actual access to me, I do a 2-hour live web class every single week. I do, that's all for my subscribers. So, okay, I want some answers, guys, before I'm not just going to give it to you. Otherwise, there's no value in it. You've got to use your brain. I'm not going to be sitting with you in the exam. So give me some answers. What do we have here? What is dwarf? The genotype or a phenotype? If you're not going to tell me, I'm just going to pack up and go home. I'm going to go plan my web class for my subscribers. They answer all my questions. I'm asking you, the question is, what is dwarf? Is that phenotype or a genotype? It's a phenotype, exactly. So what genotype is it therefore? It is Q squared equals 0.26. Because to be dwarf, you must be little t. There's only one possible option. So Q squared is 0.26. So to find Q, you'd have to square root 0.26, which I have my calculator. I've used my calculator twice today. I'm very glad that I bought it. Square root 0.26 gives us Q, which gives us 0.5. You'd keep all your digits on your calculators at this point. That's enough detail for me. I made up the question. They won't probably give you one like this. So then we can find P by doing 1 minus Q, which is 1 minus 0.5099. 1 minus my answer is, so P equals 0.4900. So 2PQ is 2 times 0.4900 times by 0.5099, which equals 2PQ, which equals 2 times answer times 0.5. 49%. 4998. What do I give this to? If this was in the exam, I would give this to, I think, 2DP. It's 0.5, so that's 50%. I just made it up. Okay, let's stop my screen sharing. It's time to go. If you've got any other questions, then feel free to save them up for next week or try out, I just try the course. If you're struggling, if you're panicking about your A-level biology, then you can try out the entire course where I teach you the entire spec. I'm sure loads of you guys are already like Naomi and Courtney, and lots of people are already subscribers. Okay, so what I was just making sure I haven't missed any. Some requests for cell mediation human response. I mean, there's a huge amount of stuff to cover there. That's more than half an hour on its own. Devise questions, then mostly going to be devise investigation. I covered that at the start, so go back and watch the recording of this. Conservation and farming, you can bring it along next week, Finn, if you want. Is 6 weeks enough time for practicing questions to go from a C to an A? If you work really hard, definitely, then there's enough. 2 grades is definitely doable, for sure. I'm actually putting a web class together. It's like how to improve at least a grade in a month. So keep your eyes open for that. Okay. We ran a promotion and the promotion is over, Sophie. So there's like, yeah, the course is all available on the site though. How do you subscribe to the web class? Well, once you're one of our subscribers, go to the My Courses page. You'll see the biology web classes course. If that's the course that you're enrolled in, you can just register for the web class, but you have to register 24 hours in advance so we can check your email address and accept you. Pierre, you're in my classes most weeks. I know who you are. I will, if you register, then I will accept you. Well, thank you for saying that. Seriously, anyone who's bought the course yet, it's a worthwhile investment in your future. Yeah, it's rad. It works. Okay, sweet. I think that's about it. Okay, so yeah, that's going to be it for the day. See you next week, or see you in half an hour, or 20 minutes. I best get my act together. I'm going to pull some questions in so I can do some. I'm going to be going through a bit more intense questions for my web class starting soon. So see you next week, and if not, then we'll be back on the Monday check-in. We didn't do a Monday check-in this week because it was Easter and we were all in different parts of the country and no one had their equipment with them. So yeah, we thought we'd just give you guys a rest, and we'll be back at the Monday check-in at 6 o'clock on Monday if you want to catch anything else from us. See you soon.